go. Okay, let's go ahead and get started. And I have a new toy. I got a new microphone. So I hope everybody online can hear me better. This is supposed to give me great coverage. This is a real microphone as opposed to little things are in the camera. So we'll see how it works. Anyway. Okay. So today we're going to do second part of Unit 1. We're going to look at cells. We're going, as far as the cell structure, we're going to look at mitosis. We're going to take a look at movement across a cell membrane. And if I have time, I'm going to go back and pick up a few slides on the skin, the integumentary system. So it shouldn't take us all that long, and it's probably going to be a lot more interesting than atoms and protons, but I didn't say that. So, okay, so let's go ahead and get into this. We are looking at week 2 lab. We'll start out with the cell cycle and mitosis. And I hope I don't move the camera around too awful much. Come on, why aren't you? There we go. Okay. So our cells, and we're going to hear more about this in lecture, but the cell is the smallest unit of living things. Nothing smaller than a cell is alive. Bacteria are alive. They're cells. Funguses are alive. They are made of cells. Viruses are smaller than a cell, and viruses are not alive. When we hear people talking about killing a virus, it's an incorrect term because a virus is not alive. A virus is nothing but a strand of DNA surrounded by a coat of protein. It has no living functions or living properties. So, but a cell is something different. A cell is the smallest structure of life. Living cells only come from other living cells. And this is a very stylized model of a cell. It's also pretty big too. Let everybody in Zoom land see this. This represents all the structures inside a cell. So we have the most prominent structure you can see here is the nucleus in the center, which you saw in the lab last week when we did the when we went through all the tissues. You saw the nuclei in each one of those cells. You saw the nuclei on the examples of the cheek cells that, if we had lab on that first week, we would have done, but such as it is. But you can see there are a lot more structures inside a cell than just the nucleus and the cytoplasm. All of these plastic structures in here represent what are called organelles. Organelles are parts of a cell that function a lot like organs do in the body. They are responsible for releasing energy. That's these little things here. These are called the mitochondria. These are where we release energy for the cell to use. In the nucleus, we have the DNA is contained. These structures here are called the centrioles, and they produce the fibers that are necessary for a cell to use when it undergoes division, mitosis and cell division. So this is, a cell is a pretty busy structure. And the other thing to remember is it's three-dimensional. It's not a flat substance. It's so easy to fall in the trap of thinking this is just, everything's laid out flat. But it isn't. It is, everything wraps around the nucleus. The cell is a ball of cytoplasm, of goop. It used to be called, years ago, it was called protoplasm. If you ever find a really old textbook, back in the 1930s or 40s, it'll say protoplasm. And a lot of the stuff that's inside of a cell in those old books is left unknown because we didn't know what it was. But the contents of the cell, the cytoplasm, is often called the cell sap or cytosol. It's sort of a goopy, syrupy mixture in here. Now let's get back. Let's leave it on that, and I can go back to PowerPoint. So we have lots of cells in our body. We have billions of cells. We have many specialized types of cells. We went through epithelial cells. We went through muscle cells. We went through nerve cells. And we went through all the connective tissue cells. Some cells can be very, very tiny, and some can be extraordinarily long. The longest cells in our body are found in a nerve that runs from our hip all the way down to our big toe. It's called the sciatic nerve. It's about this. The nerve itself is about as big as your finger. It's made up of tens of thousands of individual nerve cells. The axons, the extensions of the nerve cell, run all the way down there as one cell. So they are the longest or largest. I'm going to sneeze and it just stopped. Okay. So anyway, those nerve cells can be 3 or 4 feet long, depending on how long your nerve is, because every nerve is made up of thousands and thousands of individual nerve cells that run the length of the nerve itself. So the sciatic nerve, which starts back here in the hip and goes down behind your knee and goes all the way to your big toe, is again about as big as your finger, made up of tens of thousands of individual nerve cells. So those are the longest nerves in our body. Others are very tiny. We have red blood cells that carry oxygen. We have white cells that fight infection. We have all those little columnar cells, and we have those little simple squamous cells that make up the inside of our lungs, so we can have movement of oxygen across the membranes there. But they all have similar properties. They're all going to have a nucleus. They're all going to have a cytoplasm. They're all going to have various organelles. They're going to have mitochondria. They're going to have centrioles. They're going to have endoplasmic reticulum, series of tubes through here. They're all going to have the same structures. The only ones that are different are going to be red blood cells, because red blood cells don't have any of that. Red blood cells don't have a nucleus. Red blood cells only have cytoplasm, because they're too busy carrying oxygen, and they don't reproduce, so they don't really need to worry about any of that stuff. So they're sort of the outlier in this, in all of this. So here's a typical cell right here. Every cell except red blood cells, are going to have a nucleus, a membrane, and cytoplasm. Now, the red blood cell had a nucleus, but before it gets released from our bone marrow, that's where we make our red blood cells. Before we release the red blood cells out, they kick their nucleus out, and all the organelles go with it. So they don't have anything inside there. There's no DNA, there's no nucleus, there's no mitochondria. They don't need any of that. They just carry oxygen. That's all they do. But all the other cells are going to have the same structures. They're going to have a nucleus, a cell membrane. They're going to have cytoplasm. They may have cilia. The cells that line our airway and line the fallopian tube are going to have cilia. They're all going to have a series of structures called the endoplasmic reticulum that surround our nucleus. The nucleus is this big purple ball here. And I think I got carried away with myself again. The big purple ball is the nucleus. Inside the nucleus is the DNA. What we call the chromatin is the DNA combined with a protein called histone wrapped around there for stability. They're all going to have mitochondria. Whether the cell divides or not, if it ever divides or not, doesn't matter. Muscle cells never divide. Muscle cells, we're stuck with the muscle cells we have at birth. They can get bigger. They're going to grow when we go through puberty and hit adulthood. We can make our muscle cells bigger through exercise and eating protein, or we can make them smaller by not exercising, but we're not going to grow new ones. The same with nerves. we don't grow new nerves. If they get damaged or destroyed, they're gone. Same with muscle cells, and that becomes particularly apparent in the heart. A person has heart disease, they have a heart attack, they have damage to their heart. They're not going to grow new heart cells, heart muscle cells. They are going to be stuck with what they've got. If they have 80-20, if they have 20% damage, that means they killed off 20% of their cells. And all they're going to have, 20% of their heart's going to be scar tissue that doesn't do anything, and the rest is going to be heart muscle. So our cells are really complicated. They have lots of interesting things going on in them as to what they can do. We're going to cover a lot of that when we get into lecture. But today, just to give you a highlight of some of the structures here, the nucleus is probably the most important structure in the cell. That's where our DNA is located. And every cell has the exact same DNA as every other cell. It doesn't matter if it's a skin cell or a muscle cell or a nerve cell or a liver cell or a white blood cell. They all have the same DNA. The thing that makes them different is the parts of our DNA that make them a white blood cell are turned on and everything else is turned off. We don't need to worry about being a nerve cell because we're not a nerve cell. We are a white blood cell. So all the functions of a nerve cell are not there, are there, but they're not turned on. Skin cells are skin cells because that's the section of the DNA that's turned on for them. Liver cells are liver cells because they function in the liver, and the DNA that controls the liver cell is turned on for them. But all the other information about how to be a skin cell or a bone cell or a muscle cell, it's all there. It's just not being used. And so we don't, while all every nucleus is the same, containing all the DNA, what makes a cell unique is what gets turned on and turned off. This is how, and this, the whole, we've seen this demonstrated when we've seen cloning taking place. An animal gets cloned. They don't always use the DNA from an egg cell. They use the DNA from any cell in the body, but they find ways to turn it on so it can, so the animal can grow. The very first sheep ever cloned was named Dolly, and for Dolly, it took 3 sheep. It was like the 250th try to get a successful clone, and they took an egg cell from one sheep, and they took the nucleus of a mammary gland cell from another sheep. Now, the reason they took the nucleus from a cell, the mammary gland, it was probably easy to get to because it's probably a large cell, but because if you take a cell from your body other than an egg or a sperm cell, it's going to have the complete complement of chromosomes. Egg cells only have half the number of chromosomes. It only has 23 chromosomes. Sperm cells only have 23 chromosomes. They're incomplete. Every other cell in our body has 46 chromosomes. So in the case of the sheep, I don't know how many chromosomes a sheep has, but they couldn't use an egg cell in the sheep because it would only have half. So they took a cell from the mammary gland of the sheep, but it took the nucleus out of that, put it in the egg, took out the old nucleus, which was only halfway there, or half full, put in a new nucleus, hit it with a jolt of electricity. I don't make this up. I don't think they ran around saying it's alive, but maybe they did. Who knows? And then they implanted that in the uterus of the third sheep, and 200 and some days later, they had a new sheep that was genetically identical to the sheep they took the mammary gland nucleus from. So we know that every nucleus contains all of our DNA that can be turned on to do what we want it to do. Now, the downside is we don't know how to do that. We know how to do it to clone, but we don't know how to repair things like damaged nerves or damaged hearts and things like that. How do you turn it on to repair it? Don't know that yet. but we do know that every nucleus, the DNA in every nucleus is exactly the same as all the other cells in our body. Even though the cells look different and do different things, all the information's there. Anyway, enough about that. So the nucleus is the control center. The nucleus controls everything about the cell. The DNA, all of our DNA is in the nucleus. Every cell has a complete copy of the DNA. Depending on what the cell is the DNA is going to be turned on, or some parts will be turned on or turned off. When the cell is busy doing cell things, and the cell runs through what we call a cell cycle. When a cell is busy doing cell stuff, busy being a skin cell or a liver cell or whatever, the DNA is scattered throughout the nucleus, wrapped around this protein called histone, and we call this chromatin. You can't see it. It's because it's so tiny. But when the cell decides it's time to undergo mitosis to replicate itself, that's when the chromatin condenses and we get the chromosomes that we're familiar with. We've all probably seen pictures of cells undergoing mitosis and cell division. So it's only when the cell is getting ready to divide do we see our chromosomes. They condense then. And the only reason they condense is because each strand of DNA is about 6 feet long, and it's thinner than a thread. It's much thinner than a thread. And moving the DNA when a cell divides and undergoes mitosis and divides, you got to move chromosomes from one side of the cell to the other. And these things are really fragile. So they condense into thick chromosomes so you can move a whole bunch of DNA at the same time without damaging it. So, and that's why the chromosomes form. Okay. Now a cell runs under a very specific cycle. We call it the cell cycle, clever name for it. Mitosis is one part of that cycle. Mitosis is the very brief section of the cell cycle. Mitosis takes place in seconds. And the rest of the time, the cell is in what we call interphase. I know I can remember being taught at some point that the stages of mitosis are interphase, prophase, metaphase, anaphase, and telophase. It's easy to remember it that way, but interphase is what a cell is doing when it's being a cell. After it's replicated itself the previous time, and then it starts doing things like secreting mucus and cell sap or whatever it's doing, making proteins, that's interphase. That's its regular activity. Interphase can last for 2 days, 4 days, it can last for a lifetime. That's interphase. So the two stages of the cell cycle are mitosis and interphase. And mitosis is, if the cell is going to be able to replicate, it's always a very short time period. This is interphase. This is what we usually see when we look at a slide of a cell. This is looks like a cuboidal cell. You see the nucleus, and you can see the cytoplasm, and you can see the edge of the membrane. And that's all. But the cell isn't doing anything, or it appears to be, but it's probably very busy. But this is the cell when it's normal functioning activity. 99% or 99.99% of a cell's life is spent in interphase, doing cell stuff, making proteins, destroying bacteria, whatever it does. It's only when we get ready to divide do we go into mitosis. And this is how fast it takes place. This is real-time imagery of the process of mitosis and cell division. Mitosis, we replicate the chromosomes before we get to mitosis, and then we pulling them apart to either side is mitosis, and then we still have to separate. The process of separating the cell is known as cytokinesis, and you can see in the imagery here that the cells, once the chromosomes are pulled apart to either side, we divide down the middle. That's how we separate out. Mitosis and cell division are two separate things. Mitosis is nuclear replication, copying the nucleus. Make sure you remember that, because I'm sure there's a test question somewhere out there in your future about mitosis. Mitosis is always nuclear replication. You see, whatever the questions are, whatever the answers are on that test question, like what is mitosis? Don't say cell division, because that'll be wrong. Even though that's what we all learned early on. I can still remember in high school being taught it's cell division. Well, actually, no, it's not. At least at Walter State, it's not. So make sure that you always mark nuclear replication, not cell division in there. So, but you can see just how fast these cells divide. If a cell lives for 48 hours, its last 10 seconds or so is spent in mitosis and division. It doesn't take a whole lot of time. In lecture, you're going to see the cell cycle drawn out, and it's going to make it look like mitosis is 1/4 of the whole time cells in the cycle. And that's not true, because it's very, very brief. Because why waste your time dividing when you can be out doing cell things and protecting the body? So, okay. The 4 stages of mitosis are prophase, metaphase, anaphase, and telophase. Prophase, the chromosomes are going to condense. Remember, if you go back here and look at, well, here's a nucleus here right above the one with the red circle. Let me see if I can get that too. Come on, there we go. This cell here is in interphase. There's the nucleus. There's another one in interphase right there. But this one is in prophase. I gotta get a different color. Let's try that instead. Let's see what that does. Yeah, that's better. Okay, so here's another one in interphase there and there. Prophase is down here because the chromosomes are now visible. You can't see the chromosomes in that nucleus in either one of those, but you can see the chromosomes there because they're condensing. So the when the cell is ready to undergo mitosis, the chromosomes will condense. They will come together because it's going to be very difficult to move those chromosomes apart in the cytoplasm without damaging them. So they condense, so they're a much thicker structure. Now, where did what we do here is we've already copied our DNA. We make a perfect copy of our DNA earlier in the cell cycle and just sort of park it there till we're ready to go. But we condense our chromosomes. We see the nuclear membrane, the membrane around the nucleus itself disappears. And those little centriole structures, these things that I showed you on the model over here, these are called centrioles. Every cell has a set of centrioles like this. Well, they've copied themselves too, and one set stays here, and the other set goes to the other side. And this is where your spindle fibers, you've all heard the term spindle fibers, right? At some point, the spindle fibers are going to grow from centrioles on both sides. So we see these in place. That's what happens during prophase. So our nuclear membrane's gone. The chromosomes are visible, and the centrioles have decided it's time to migrate to both sides. Second stage is the most obvious, and that is called metaphase. Metaphase, all the nuclear structures are gone. The chromosomes have moved to the middle of the cell. They've lined up on the what we call the equatorial plate. That's an imaginary line right through the middle of the cell. And the spindle fibers are attaching to the chromosomes. Now, some of them attach to the chromosomes and some don't. Some just extend out into the cytoplasm, because not only will the spindle fibers help to move chromosomes to each end of the cell, they will also push the cytoplasm apart, because that's how that helps get our chromosomes on either end. So metaphase, very distinctive. You can see on the red slide, you can see there they are lined up in the middle of the cell. And even over here in the one that's circled, they're lined up very, they're all together. Now, the reason they do that is so that we can make sure that no mistakes happen, that we get the same number of chromosomes going to the left and going to the right. In a human cell, we have 23 pairs of chromosomes. We have 46 chromosomes. We want to make sure both new daughter cells are going to get all 46 chromosomes. We've copied the DNA. We want to make sure there's no mistakes. In lecture, I'll explain why mistakes happen all the time, but we can fix them. So, but for right now, we want to make sure there's no mistakes. Okay. The third phase of mitosis is anaphase. This is the shortest stage. This is when the chromosomes actually start to move. They move from the middle of the cell to the parent cell to either side of the cell. Right now, you've got a large cell with lots of cytoplasm and two complete sets of chromosomes. And the reason we do this so quickly is we don't want to have 92 chromosomes in a cell trying to tell the cell what to do. We want to get that done quickly. So that's why it takes only seconds. The spindles pull the chromosomes apart, they push the cytoplasm apart, and we are just about ready to go to the last phase. Just let me show you just how quickly, you can see just how quickly the chromosomes are moving here again. I mean, there they go. This one here really shows you anaphase, how fast it takes place. When you see this one, how very quickly anaphase moving to the last stage occurs. So let's go back up here. The last stage is telophase. Our chromosomes have moved to opposite ends of the cell. If nothing else happened, if we stopped right here, we'd have a very large cell with two complete sets of chromosomes. We have to separate the cell. Now, while the chromosomes move over in the either end of the cell, they start to unwind. They start to separate out into the individual structures instead of being condensed. They start forming the new nuclear membrane and nuclear structure. And this process known as cytokinesis. Cytokinesis starts in anaphase. Cytokinesis starts here and continues into telophase and finishes. And then, so you can start seeing this little line right here. You definitely see the separation here. What happens is that there's a band of protein around the outside of the cell. And that band of protein, it's the same type of protein we have in our skeletal muscle. It's a contractile protein, and it starts to squeeze. And it contracts on itself and contracts on itself. And it squeezes it and squeezes it and squeezes it until it pinches off. You ever see one of those balloon animals that kids get? How they twist it and they can make all the shapes. That's sort of what happens here. You're squeezing the membrane of this very large cell, and it doesn't pop. It just comes apart into two new daughter cells that are identical. And so we end up, this, you can't see it, but the band of protein is right here, causing this to separate. Cytokinesis has to happen to give us the two new cells. Without cytokinesis, we just get one large cell. So that's why we say mitosis and cell division. Because without the cell division part, without the cytokinesis, we don't get two daughter cells. Here we can see This line here is called a cleavage furrow, where the got carried away with myself again. There we go. This line right here is the cleavage furrow, where the protein has continued to squeeze and squeeze and squeeze the cytoplasm to cause it to separate. And we end up with two distinct daughter cells. Each brand new cell has a complete set of the chromosomes that we started with. Now, so then we have two distinct daughter cells, and this will go on for however long the lifetime of the cell is. Some cells live for a couple days, some cells live for a couple weeks before they undergo mitosis again. Some cells never undergo mitosis. Muscle cells and nerve cells never go through this. Red blood cells don't undergo mitosis. They last for 4 months and then they're replaced. White blood cells some will last for days, some will last for a lifetime, because we have white blood cells that contain memory on how to fight infections. So that is mitosis. Now let's move on here and talk about movement of fluids across a cell membrane. Sounds really exciting. So, but it actually is pretty important to us. When we get into AP2, we'll spend a lot of time talking about movement across membranes. There's a fluid called interstitial fluid. Interstitial fluid is plasma. Plasma is the fluid that our blood carries our cells in. Red cells and white cells and platelets are carried in the plasma. Plasma is salt water, and the red cells and the white cells and the plasma float in it. The red cells are there's so many red cells in a cubic milliliter of blood that that's why it makes it look bright red. But there's white cells there too. The plasma leaks out of our blood vessels. Our blood vessels are not a solid tube. They've got lots of openings there, lots of holes in there. The plasma is always leaking out. It's oozing out into the cells around it. Excuse me. So here's a cell right here. The plasma is leaking out around those cells, and it's here's your cell. Plasma is leaked out of the blood vessels, and it surrounds the outside of the cell. We call that interstitial fluid. It was plasma. It's made of salt, water, and proteins, and it's like 99% water, and some salt and protein in it. Surrounds the outside of the cell. We call it interstitial cell. It is the same concentration of the water inside the cell. So water's out here, water's in here, and this water surrounds the cell, and then eventually we recover it back into the plasma. So, but it's important because that fluid, that interstitial fluid, is used to give us a way to monitor for infections. Anyway, so the cells are surrounded by interstitial fluid. Inner means outside, intra means within. So it's not water inside the cells, it's water around the cells. The cells use that interstitial fluid because that's where they're going to pick up ions of salt. They're going to pick up glucose. They're going to pick up hormones are going to come into the cells through this interstitial fluid, because the hormones in our bodies, the chemicals that, like growth hormone, for example, they're going to float in the plasma and they're going to ooze out around the cells. They're going to move into the cells, or any sugar that's floating in the plasma is going to be on the outside of the cell membrane, and it's going to work. It's going to find a way to get in. Cells depend on the interstitial fluid to pick up nutrients, to pick up oxygen, to pick up proteins, to pick up hormones, and to get rid of waste products. See, our blood vessels are carrying all of our nutrients in solution. They're carrying our oxygen in solution. The oxygen's being carried by the red cells. And when the red cells reach, are in a blood vessel next to a cell. Here's a cell, we got a blood vessel next to it. For that oxygen to get into the cell, it has to leave the red blood cell, go into solution in the plasma, cross the membrane of the vessel, cross the membrane of the cell itself before it can go in, and it has to stay dissolved in water the whole time. So that's where the interstitial fluid helps us. Movement across a membrane is passive. It usually doesn't cost the cell anything. Movement of oxygen into a cell is free. We all like free, right? Who doesn't like free? Free is a good thing. Oxygen moves into our cells freely. It's a passive process. Carbon dioxide leaves the cells passively. We don't have to do anything to get rid of the CO2. It moves through a process known as diffusion. The nutrients, glucose, will come into a cell. Sometimes it needs a little help, but it's still passive. It's a free ride. We don't have to spend energy to get glucose into our cells. It's only when we have very large molecules that we need, or that that we have to work extra hard to get them in there, that we have to spend any kind of energy in here. So, because our cells are cheap. They don't want to spend anything, but they have to in some stuff. It's probably not a good technical term. Cells are cheap. Cells don't like to waste energy moving stuff across their membrane. So, but most of our transport across a membrane is passive. Passive transport, free ride. Like water flowing downhill, that's passive. Water flowing uphill is active. Active transport takes energy. Water doesn't flow uphill. We all know that, right? So, unless we have an earthquake, then it does. But that doesn't happen that often. So, water flows downhill passively. It's a free ride. However, in Tennessee, there's a place, I think in Chattanooga, where TVA will pump water up a mountainside at night, store it in a reservoir, so it can run back down during the day and turn the turbines in a generator. And it costs less to pump it up at night when there's low demand, so it makes more energy during the day when it comes back down. It's sort of like an emergency reserve. So, but how did you get the water uphill? You had to pump it. You had to pump up the hill. That took energy. Apparently they get more back out of it coming back down, but I don't know. I'm not an engineer. So, but our cells will often have to spend energy to move things inside or outside. That is difficult. So, okay, moving on. Cell transport, membrane transport. We see three different ways here. Diffusion, simple diffusion. If I put a drop of food coloring in a beaker of water, the food coloring is going to spread throughout. That's simple diffusion. It just moves out on its own. There's nothing I can do to make it to stop it. It will just, the movement of the water molecules will move the food coloring around through the beaker. That's simple diffusion. In our cells, substances can diffuse across this layer here, the cell membrane. Oxygen can diffuse across the membrane very easily. Oxygen comes into the cell passively. Carbon dioxide leaves the cell passively, right across the membrane. It's the ideal arrangement for those gases. Facilitated diffusion requires some help. Water can't go into a cell on its own. The cell membrane is made of fat. It's what we call a phospholipid bilayer. I'll get into that in the lecture, but it's a fat layer. And oil and water don't mix. We can't add, water won't go through an oily layer. So but we have channels in the membrane that allow water to go through. It's sort of like a tunnel, through or a pipe through the membrane. Water gets in. Doesn't cost anything. We call it facilitated diffusion. We help water get into the cell through these channels. Glucose comes in the same way. Glucose comes in through facilitated diffusion. Glucose is a large molecule, and it is not dissolved, as we say, fat soluble. So it's not going to slide through the membrane very easily. But there are channels that can open up in the cell to let glucose come through. That's what insulin does. Insulin, I want to trip over the wire. Insulin opens up channels in the membrane of a cell so the cell can absorb glucose. Glucose is too big to diffuse, and it wouldn't get across to the fat layer anyway. But by having a special channel for it, we get all the glucose we want. That's what insulin does for us. It allows us to take in the nutrients we need at the cellular level. Now, the third mechanism, though, is the one that takes 40% of a cell's energy budget. Active transport means that we have to spend energy in the form of a molecule called ATP to get something to move across the membrane. Now, you'll notice that on the first two, both of them were types of diffusion. Diffusion through the layer, like gases. Facilitated diffusion through a channel. Again, notice that you're going from a high concentration to a low concentration. On the first one, those black circles might represent oxygen. And so there's a high oxygen on the outside of the cell and low oxygen on the inside. So oxygen just moves passively through diffusion from a high concentration to a low concentration. The same with the facilitated diffusion. Those orange triangles might be glucose. More glucose outside than inside, so glucose flows in. But on the third one, you can see that the high concentration of something, these green squares, there's many of those in here. So there's a higher concentration inside the cell than there is outside. And yet we want that stuff that's outside. It's not going to move into the cell passively because the concentration gradient is high on the inside, low on the outside. The only way the concentration gradient would work for stuff to leave. So we actually have to pump those little green squares, and then we have to actually pump them in. We don't want to lose those green squares. Those green squares could be potassium. We have a great need for potassium in our cells. So we pump the potassium in and we spend energy to do that. Active transport always requires energy. It's always moving against the concentration gradient. It's like pumping things uphill. So, and I still don't know how TVA can make any money pumping water at night, letting it come back down, but it really does. Okay. So I said, transport across the membrane is either passive or active. If it's passive, it's free, there's no energy. It's almost like gravity. I mean, it isn't gravity, but it's a free ride simply from a high concentration to a low concentration. Active transport moves against the gradient. It's like moving uphill, and you have to use energy. You have to use ATP, the chemical that we use as our coinage to pay the price, if you will. Now, I've already mentioned this in lecture about solvents and solutions. Ideally, solutions can would like to come across the cell membrane freely. Sometimes they do, and sometimes they don't. We know what a solution is. It's a homogeneous mixture, meaning that you can't look at a solution and say, oh, there's the sugar, there's the tea, there's the water. It's all mixed together. When you look at a Coke, you can't see the caffeine, you can't see the sugar, the high fructose corn syrup, you can't see all the other ingredients in there. that have to come together to dissolve the sugar and give you the flavoring agents. It all looks the same, and it's a solution because it's transparent. Even though the caramel coloring there makes it look dark brown, you can still see through it. Now, whatever the dissolving medium is the solvent. Whatever is being dissolved is the solute. So let's just think of salt water. Water is the solvent, which it usually is in the body, and salt, sodium chloride, is the solute. We dissolve it in water. Now we have water and salt outside our cell. We have water and salt inside our cell. We have intracellular fluid. That's the water and the cytoplasm inside the cell, and we have the interstitial fluid. That's the water on the outside of the cell. As I said, the cells are going to be picky. They're going to restrict movement. Things that are dissolved, things that are fat soluble come across very easily. Things that are water soluble have a hard time. That's why we have to have channels and things like that. We say that the cell membrane is selectively permeable as to what it comes in, what it allows to come into the cell and what it allows to leave the cell. You know, the membrane functions like our skin. It protects the cell from stuff going in or stuff going out. The cell doesn't want to get too dehydrated, and it doesn't want to become overhydrated. You know, too much water into a cell will cause the cell to pop. Too little water will cause the cell to shrink up. In both cases, they become non-functional, particularly if it pops. So diffusion, simplest process here. Passive transport. Here we have a dye pellet dropped into a beaker of water, and you can see how it has spread throughout the beaker, and it turns the whole beaker purple. So, okay, we go from a high concentration where the pellets first goes in to evenly distributed. It has reached equilibrium. So simple diffusion. The definition for diffusion is movement of a substance from a high concentration to a low concentration, just like it says there. It's random. The water molecules are moving, the dye substance is moving, they're all bouncing around, and the movement of the water molecules gradually moves the dye or whatever material we have until, excuse me, until we reach equilibrium. Totally passive, no energy involved. The random movement of the water molecules doesn't count because water molecules are always moving. So simple diffusion from a high to low. Now, if a membrane gets in the way, we have an issue. Because the only thing that can diffuse simply across a membrane is something that's fat soluble, or if it's so tiny that the membrane doesn't care. Gases don't care. Gases are so tiny, the individual molecules of gases are so tiny, they just slide right through. But water can't go through a cell membrane because it's not soluble in fat. So gases are small enough to go through the membrane, water can't. So, but water gets in because of a carrier molecule, a channel, if you will, a protein. It will slide through and give you what you need. It'll go in, water will go in through the carrier protein, insulin goes in, insulin builds carrier protein so glucose can go into the cell. And so we bypass, we're bypassing the phospholipid layer so we don't stop it. So, anyway, simple diffusion doesn't need any carrier proteins. Facilitated diffusion needs them. Okay. So simple diffusion is unassisted. I said it already. Passive transport. Now, what if you have something that's fat soluble? Certain of our hormones are made out of cholesterol. They're a steroid. They're fat soluble. They can slide right across the membrane. The sex hormones are fat soluble. They don't get hung up on the membrane. The testosterone and estrogen can go into cells very easily because they're fat soluble. Any steroid hormone, and there are quite a few of them, actually, can go right across the cell membrane with no trouble. They don't need any assistance. A large hormone node that's made out of protein can't get through. It's not fat soluble. So, now here's a term, and I've repeated myself a couple times here on purpose. Here's a term that's very important to us: osmosis. Osmosis is the movement of water across a cell membrane. It's still passive. It's a type of diffusion. We go across the cell membrane from a region of what we call high water to low water. High water to low water. So we have a membrane. Here's our membrane. Here's the outside of the cell. Here's the inside of the cell. And of course, the membrane is a lipid layer. But whatever the membrane is, we will have channels in here for water to go through. There's a water channel there, and a water channel there, and a water channel there. Now, when I say high water and low water, suppose you have a salt solution that is 10% salt and 90% water. So 10% salt solution. We'll put 10% salt inside here. 10% salt is solution is 90% water. What if you place this cell in distilled water? Distilled water, DW, is 100% water. So we have a cell with a member. You have a cell here, and inside the cell, it is 90% water, 10% salt. Outside the cell, it's 100% water. We know that osmosis is movement from high water to low water. Which on the what's the water percent here on the outside the cell? 100%, right? Thank you. And the water concentrate percent inside the cell, which is the high water side, which has a higher water amount. The outside, the 100%. Water will go from the cell, from the outside of the cell in here, and the cell will enlarge. Water always goes from the high water side to the low water side. So if you put a living cell into distilled water, that living cell will enlarge. It will swell up and maybe even rupture. Red blood cells will pop all the time in distilled water. So we have 100% water is greater than 90% water, so the water goes into the cell. Let's change the parameters a little bit and change this. So all we're leaving behind now, let's say that the inside of the cell is 90%. Oops. There we go. Okay. Let's say the inside of the cell is 20% sodium chloride, which means it's 80% water. The outside, and we'll put the outside in a 40% sodium chloride, which is 60% water. Which is the high side now, the outside or the inside? The inside. Which way is the water going to go? It's going to go out like this, and the cell's going to lose water. Osmosis always works. It always goes from the high water side to the low water side. Doesn't matter what the solute is. It can be glucose. I could have made that a 40% glucose solution on the outside. it still would have had more solute particles than the inside. So water would have left the cell. If you, and this is what happened, you know, in the first example, where you had to put a cell in 100% water solution, this is what happens when someone overhydrates. You know, where someone is very, very thirsty and they drink a whole lot of water at once. There's a condition that occurs if, like, let's say that you've run a marathon, and at the end of the marathon, you're so thirsty, you drink 6 liters of water, right, one after another after another. It's possible. You dilute the salt concentration in your plasma and in your interstitial fluid. The amount of salt in our body is pretty constant. We don't lose or gain too much salt, but we can lose or gain water. So you just chug down 6 liters of water in about 2 minutes. You were really thirsty, you know, it's a hot day, you know. And so you chug down all this water. Where's all that water goes? It goes into your blood. Everything we eat and drink goes into our blood. So you suddenly are diluting the salt concentration inside your plasma, which means that the salt concentration I'm getting that backwards. Hang on a second. Let me continue with this so I don't confuse you. So you've diluted the plasma, you've diluted the salt in the interstitial fluid, and so water leaves the cell and goes out, and the cell shrinks because you've got all this extra water here. In this case, suppose that you're dehydrated. Suppose that you have a patient who has been drinking heavily. One of the consequences of drinking heavily is a need to urinate a lot, because alcohol interferes with the kidney's ability to regulate water levels. So a person from drinking heavily and they've been peeing out a lot of water and they become dehydrated. So you have this person who's dehydrated. Well, what do they lose when they're dehydrated? They lose water, they don't lose salt. When you're dehydrated, you sweat or you pee a lot and you lose your water volume, okay? Or maybe this person's been sitting outside in the sun for 3 days and not drinking water. I don't know why, but you know, they're not getting the water they need. They've lost water. But when we lose water, our salt level goes up. Because we don't lose salt. When our water level drops, our salt concentration goes up because the salt stays there. Let me go back to my beaker. If I have a beaker, where do I put it? Oh, here it is. If I take a beaker and I put 100 grams of salt in here, and I put a liter of water in here, I've got a 10% salt solution. Everybody with me in this? 10% salt solution. So I let it set out and I let it evaporate. And I still have 100 grams of salt in here, but after a week, I'm down to a half a liter. Now I have 500 mils of water, and I still have 100 grams of salt because the salt hasn't gone anywhere. The concentration here now isn't 10%, it's 20%, right? Because you know, instead of 1,000 mils, I only have 500 mils, and 100 grams is 20% of 500. And what if I let it evaporate again down to 250? Now I'm at like, what, 40% salt? I haven't done anything with the salt. It's still 100 grams, but the volume of water has been dropping. And so the water's been getting saltier and saltier and saltier as we lose, you know, as the water leaves and the salt's left behind. In our bodies, if we lose water, the outside of the plasma and the fluid around our cells will get saltier. And so our body's response is, well, if we're getting dehydrated, the cells are going to give up water to the outside. The water inside the plasma, inside the cell membrane, water inside the cells is going to move across the membrane from osmosis. Because remember, osmosis is higher water to lower water. And so if we lose enough water outside the cells, we only have 60% water. Here we have 80%. The cell's going to give up its water and shrink. We're going to go into great detail on that when we get into AMP2, because those are 2 good ways to kill your patient. You know, to get them dehydrated or to overhydrate it. People can die from water intoxication just as easily as they can from dehydration. Too much water or too little water is going to affect the body. So osmosis is really important. We don't think about it a lot, because osmosis, normally we don't worry about the salt concentration being different outside the cell or inside. It's not something, I wonder what my salt concentration is today. I'm so worried about it. We don't think about it. It takes care of itself unless we get into some sort of situation where we're low on water or high on salt. And our bodies are very sensitive to salt. We get thirsty when the water level drops 1%. That's all it takes to activate. Our bodies don't measure salt. I'm sorry, our bodies don't measure water levels because that'd be too hard to do. But it's very easy to measure salt concentration. So we measure salt concentration and we infer, our body infers the water level. And if our salt concentration goes up 1%, we get thirsty. We have to have something to drink. If it goes down 1%, then we have to pee, because that means we're overhydrated because our salt levels are pretty constant. And so and the only reason that the salt level would go up or down is because we're diluting it more. Take my beaker here with a liter of water. If there was a bigger beaker and I added a second liter of water to it, now instead of a 10% salt solution, I'd have a 5% salt solution. Still only 100 grams. We didn't do anything to the salt. It just got diluted or concentrated depending on what happens to the water. So that was I created my own rabbit hole in that one. So basically what you have to remember is osmosis is movement of water across the membrane from high water to low water. That always works. As long as you know the percentages of the solutions outside and inside, water goes across the membrane from the high water side to the low water side. Oops, nice way I erased it. There we go. Okay, yeah. This is what happens when you give me a toy to play with. 2 years without markers on the whiteboard, and I don't know how to control myself now. So, okay, moving on. This is simple osmosis. Again, this is a membrane, and we see the little blue circles are water, and we see the sugar is green, and the high water side is on the left, and more water goes into the right side because the left side has low sugar, high water, and the right side has high sugar, low water. We always go from the high water side to the low water side. And it doesn't matter whether it's a liquid or a solid or a gel, we can move through membranes. This is not osmosis, this is just diffusion. We can take a petri dish with nutrient agar on there. It's sort of like Jell-O. And if we drop crystals of two different chemicals, one is methylene blue and the other is potassium permanganate, the size of the molecule of the chemical determines how fast it will move through the gel. Small molecules move faster than big molecules. So you can look at that and say, well, potassium permanganate must be smaller than methylene blue because it's moving faster. They were dropped together at the same time, and the brown potassium permanganate is moving rapidly across the gel. Methylene blue is a larger molecule. It takes longer to work through the gel. So one of the ways that diffusion is affected is by the size of the molecule. Little molecules move through diffusion faster than large ones. Little ones go through faster than large ones. So size matters. In diffusion, smaller is better. We'll just skip past that. Okay, actually, that covers everything I want to talk about. Any questions on this? I know I've thrown a lot at you, and you're going to hear me talk about it in lecture when I have a little more time to give you some better examples on it too. Any questions on diffusion and osmosis? Diffusion is just movement from down the concentration gradient from a high concentration to a low concentration. It's passive. Osmosis is movement across the membrane from high water to low water. It's also passive. They always work. So, okay. Anyone have any questions on anything? So, okay, Thursday. Yes, ma'am. Oh, yes, thank you. I would have forgotten about that. You can thank her for reminding me. No, we do have a test next week scheduled. Since this is only the first full week of classes, what I may do is just slide the test back a week and start looking at bones next week. And I will give you an extra week to get it ready, because it's not really fair to say, oh, by the way, you got a test next week. So we'll move the lecture test back a week, and the lab test back a week. So that would be the 7th, 8th of February, whatever's in the schedule, we'll move it back one week. And next week we'll start out looking at bones of the body. It'll give us an extra week of bones. So, okay. So don't worry about the test. I'll send out an email to everybody to remind everybody that we're not having a test next week. So, because nobody's ready for a test, right? I'm not ready for the test. Okay, let me get out of here. Anybody have any questions online? Okay, then y'all everybody have a great